Okay. Last class we have studied about the green energy resources such as biofuel and biogas. So today we will discuss about synthetic biology and its applications. So what is synthetic biology? That means synthetic biology uh, is a new trend of utilizing our technology that is mainly due to modification of organisms. Okay. Is my slides visible to all of you?
Yes, sir.
Okay. So synthetic biology or synthetic applications of genetic engineering, it may be called as, this is the new term nowadays which can be utilized for the green energy resources. So green energy, that means we are going to be utilize or modify some things which cannot be harm anything towards an environment and easily can utilize towards our energy needs. In other words, uh, extra needs of the developing country and the more population. So synthetic biology is the new terms, or genetic, it's called also, now it's called genetic engineering, where you can use or modify gene along with its different procedures. So let's start about this, what is synthetic biology and how it nowadays it can be applicable. So definition of this synthetic biology is, it refers to use of this computer assisted biological engineering to design and construct new synthetic biological parts, devices and systems that do not exist in the nature, and redesign of existing biological organisms. That means we are going to interrupt the already existed different organisms and we're going to modify according to our needs. For example, we are going to produce more resist variety and more production of the some crops. So we need to redesign or we can be change the gene to for this particular which can be more uh, uh, that can be responsible for production of new varieties and more varieties, as well as it having a resistance to disease. So we are going to modify that particular gene of a this organisms, living organisms. So it is combination of biological engineering and computer assisted engineering. That means computer applications and biology both are included in this synthetic biology, where we can utilize our knowledge or engineering to modify according to our needs some of the biological parts such as in the gene level. And we can also develop new devices and new systems that do not exist in naturally, and these can be utilized for our needs or for better sustainable growth. So the limit resource are limited, but within the limited resource we can be find a way or utilize the technology to modify in the gene level which can be utilized. So this is the definitions where we can be utilize this synthetic biology term. The synthetic biology incorporate also techniques of molecular biology, and those who are in domain of biology and life science or genetics, they better know this molecular, what is molecular biology, and how we can DNA, how DNA, RNA that are be consisting the main parts of the DNA, RNA. What are the main parts or the sequencing of DNA, RNA, A, T, G, C, like that. So this sequencing of gene or this molecular level, that may be responsible for a particular behavior or particular traits. So in this case, this type of study or this type of modification can be done with this molecular biology level. With the term synthetic is utilized here because it can be a new versions of a existing organisms or life that cannot be existed naturally. So we are going to synthesize it accordingly. So, in other terms, the synthetic biology is related to terms of bioinformatics. Those who are having interested in bioinformatics or biology, in genetic level or molecular sequences, they can be studying further what is bioinformatics, how computer system or computer applications can be utilized to sequencing the gene, all the things. So it differs from recombinant of DNA technology that synthetic biology introduced synthetically structured parts is not limited to the modification of natural organism. That means it is not limited to only modifications, and we can also synthetically incorporate different things in our molecular level, which can be called as synthetic biology, and the construction of new life forms with no natural counterpart, because God had already created some creatures or some organisms, but synthetic is something we can create it according to our needs and according to our utilization of technology. We created this creature or these organisms which may be utilized and useful for us. So that's why it is called synthetic. The relationship between system biology and synthetic biology, what is the relationship? Because all we know, the system biology, that means the DNA, RNA, and the number of chromosomes, all the things that we cannot be modified for a particular species. So this system biology is the study of natural biological systems such as whole, often with biomedical focus, and utilizes simulation and modeling tool in comparison with the experimental information. This is called system biology. That means we are going to study the whole biological system in a computer assisted simulation and modeling tools, with these different types of DNA, RNA sequencing, all the things. And engineering design cycle and rational design cycle of synthetic biology, what is that? This is the key aspect of the synthetic biology which differentiate it from genetic engineering and current biotechnology approaches. Actually these terms are going to somehow related: genetics, molecular biology, system biology, synthetic biology, bioinformatics. These are the common terms, but little differences are there. The basic things are same, but when we are going into broad topics, we are focusing on the particular aspect of the utilizations and its productions, all the things. So that's why it is little bit different. It is saying that genetic engineering and biotechnology, these are the main basic approaches where we can utilize this approach to study the synthetic biology and its application parts. So biology techniques are normally used for this design and development. For examples, you are going to construct a house, so you have to design. According to design, you have to structure what is the need of these materials. So you have to first calculate all the things. Similarly, for modifying organisms or synthetic biology, we have to going to design what should be in a DNA and RNA molecular DNA level. So this is the engineering cycle. That means first is has having specifications, design, modeling, implementation, and testing. So we have to give a different type of specifications. For example, if you are going to develop a new organisms which is having more yield, 100% yield crop yield for one species, so that is our specifications. We are giving the traits, disease resistance variant, species, then more fruitings, more flowering, or these are our specification along according to which we are going to design. So first is specification, second is step is design. According to the given specification, we are going to modify this and design it. Then after designing, we are going for modeling. Modeling is nothing but the computational simulation model, do not exist in real world. Going to utilize this model, that means before going to a large scale productions or before going to actual, we are working in a prototype or modeling scale. So this is called modeling. If it is successful, then third step, sorry, fourth step is implementation in the real world or implementation in the real technology, real field. After implementation, whether our specifications are actually working or not, we are going to testing or going to validate. That is the fifth stage. So this is the engineering cycle. And if the specification initially designed are not working, or somehow it is not possible, or it is working, or it is going to be tested or not verified or not, if it is not, then again going to give a new specification and again going to new redesign. So this is a complete cycle process. If it is passed with the testing, then it can be okay and going for the mass production. Then Differential approach of synthetic biology. What are the approaches? How synthetic biology can be studied? First is DNA synthesis. At the most basic level, synthetic biology involves use of synthetic DNA that was uploaded or written on a computer or printed. We are cannot going to modify directly in a particular organism or in its DNA. So we have to utilize our computational resources. So the sequence or the particular DNA RNA structure is printed or written in the form of binary codes in computers for the modeling purpose, that is called the reprinted or reprinted in a DNA level. So our from the bottles of nucleic acids, that means the ATGC, that A for adenine, T for thiamine, C for cytosine and G for guanine. This ATGC are the basic nucleic acids of the any DNA RNA structures. So the sequence varies, the number of chromosome also varies for different species. So the basic nucleic acids ATGC can be structured or printed in our computer system for a modeling for synthesis of DNA. Actually, we cannot directly synthesize DNA. This is natural processes. But in the computational process, we are going to. It can be imagine anything. Anything you can imagine in computer graphics or computer. That's why in Hollywood pictures, everything is possible. That is in graphics. So like that is DNA synthesis is first done in the computer or written printed in a DNA level. This DNA stands for inserted into organisms through variety of genetic engineering technique. If the printed DNA techniques DNA is successfully done, then it can be redesigned and inserted to existing organism. So that is the first processes or approaches of synthetic biology. First have to redesign the or design the synthesis of the DNA, then incorporated or inserted to that synthesis DNA to a organisms with the help of genetic engineering. How this genetics engineering works, that you have to go for details in life science people that may be having better knowledge of genetic engineering. That are called biobricks. Biobricks are nothing but the standard DNA sequence that code for certain functions. Bricks. Bricks means the individual blocks of a house or individual blocks of any structure consisting of bricks. Similarly, for any species, we are going to specify some information and specification that is written in a computer language or written in a particular synthesized DNA. That particular DNA is you fixed for a particular functions. So this is called biobricks. So these biobricks are inserted into different organisms. So DNA sequences created make the organisms grow. And for example, engineering that biobricks into organisms could be grow. That means DNA sequence that inserted make organisms grow. That means if your organism is not growing, we are making some functions. This is the example. If you are modifying the gene level, that organisms is glow in night time, and that gene is inserted in these organisms, then obviously it works, then the organisms can be glowing at night. So these open sources bricks can be used to researchers across the world to new genes and DNA sequences. Open source bricks, these open source bricks are something which are already store in a database of the different types of international resources, which can be already, if you want some specific functions, then just we need not to again redesign, just pick up the data sources. These biobricks, each biobricks having certain names, just pick out the biobrick bricks or this DNA sequencing and utilize in your own work, that is already available. Then minimal cell or genome. Minimal cell means researchers most notably uh, Craig Ventures are working in the procedure organism or cell with the minimum number of genes needed to survive, because there are lots of number of genes are there for and if the number of gene and chromosome have increases, then the complexity also increases. So in order to reduce the complexity, it is to now define the minimum number of gene which will be organism is required. So that also be work is going on, and one could add only DNA sequence in this minimal genome or cell to produce fuel, medicines, and any other synthetic products. That means this synthetic biology is utilized for different purposes, for production of fuel, for production of medicines and other synthetic products, other cosmetics or any or anything that can be utilized to this synthetic biology to utilize to form this growing demands of this product. See, this is the structure. First, you have to design this in computer, that is in silico design in genomes. That means it is the example of a synthetic biology, what is actually it works. First, computationally you have to design, then synthesis of oligonucleotides, then according to the design you have to synthesis the oligonucleotides in a particular wet lab. Then that oligonucleotide is incorporated in the E. coli bacteria. This is an example. Then after E. coli bacteria incorporated, that complete genome assembly takes place. Then again it will be redeveloped and going to transformations takes place. So this is the procedure. How this design genome or sequencing can be implemented to this E. coli bacteria. Because first E. coli bacteria is something look like yellow, but after the incorporation it looks like a different thing. So according to our needs, we have to design this kind of things. Then next term is called xenobiology. Xenobiologists are attempting to create alternative genetic systems such as novel nucleic acid, suicide genes or mirror biology. These are also one applications of the parts of this synthetic biology. That means they are synthesize some genetic materials and new novel nucleic acids, new type of nucleic acids or genetic materials which can be utilized for alternative genetic systems, that may be called a suicide gene. That means it also having different types of the sequences that can causes the death of a particular organism, that is called suicide gene. If it is, for example, one research team has replaced thymine with 5 a colorful cell and genome E. coli, where others attempting to create mirror cells. This is one example. If the thymine is replaced with this 5C, then E. coli one it will replace the other E. coli also. They can also just clone that set or mirror that cell themselves, and they are going to replicate one another to more. So this is the difference between how this thymine is replaced with this chlorouracil. The simple thing is you have to this CH3 group is replaced by some this chloro group or chlorine group. That is the only difference. So we in our system synthetic biology we just modified this thymine as this chlorine group, uracil group. So then it will be create some mirror cells, like whenever it looks like a mirror, then it is the opposite to each other. So this is one example. So protocells are the researchers testing combination of inactives such as different synthetic life cycle without DNA. These are called protocells. In primitive eukaryotic cell you have to you have seen that there are so many nucleic acids are there, and some are testing for the different types of DNA, RNA sequencing, protein, all the things. So the protocells would be like truly uh, creating life for scratch. That means how these protocells are forming in our ancient cell of pre-eukaryotic cells are. So the origin of life should be a the questions how life origin in the earth, that can be also answerable due to these prototypes or protocells techniques. We will not go to there. Then fundamental technique of synthetic biology, how these techniques, how synthetic biology can be uh, studies. So there are three keys of technological enables that have facilitated in emergence rapid development of synthetic biology. There are first step is computational modeling, second is DNA synthesis, sequencing, and third is DNA synthesis. We already discussed these things. Computational modeling means it does not actually happens in the wet lab, but in computational we have to just modeling this sequence ATG, CGG, CTC. So this sequence are modified as sequence are modified according to our need. So see in the figure there are different types of sequencing are there, and we are just replicate one term for each of these sequences, that is ATGC, all the things. So this is the new sequencing, which is the combination of this 8 sequencing. So this new type of sequencing that is done in the computational level or computational modeling, if it is success, then DNA sequencing occurs. So the repetitive sequencing are added, then finally DNA synthesis happens in the wet lab. So computational modeling is the first step where synthetic biology approaches to design engineer biological system through engineering cycle. Modeling of the design predict system performance prior to fabrications. I already showed that computationally it was designed the synthetic, that means the DNA sequencing is the important step. If it is success, then going for the DNA sequencing. The reading of sequencing of DNA is second key to combination of synthetic biology. DNA comprises of four bases, which is A, T, G, C, and the entire content of DNA of a particular organism is called as genome. Life science people have already studied what is genome. So total sequencing of a DNA is called genome, and this contains complete instructions of the construction type of protein, cell, tissue, organ, etc. If the compositions or the sequencing ATGCGCA, this type of sequencing, that every unique, so see the large number of permutation and combinations may be happen. So it is very much difficult to identify or difficult for a sequencing the DNA, it is not easy. So this DNA sequencing is most important. If the small sequencing is changes, then whole functions of the gene can be changes. So in order to see how what is the type of gene and how it is sequences, the whole function structure, all these decision and working principles all are changes. So depending of our needs, we can sequencing the DNA. Then after successful synthesis, we are going for DNA sequencing. We are going for synthesis. One genome has a sequence. That means which what is the sequence of the genome. That is A, T, G, C. What is the next step is to be rewrite or synthesize the part of the genome. We are going to synthesize the small parts, and this synthesis part is going to be replicated for a production of large number of, we have had the name PCR, which can be also utilized for the replication of this sequencing gene. These are the number of cases where genome of the organism has been entirely synthesized. See, this is how DNA synthesis, DNA synthesis takes place. This is the different structures of DNA and the linkage. Where this, if you are unfold, that means leading strand 3 and this 5 both are one strand can be combined to form a new synthesis DNA of 3 and 5. So this is the one examples. Do not go in details. This is in 2022, 2002, Stello and workers also State New York and Boston University synthesized prolifin genome from the published sequencing producing the synthetic organisms. So the first synthetic organisms, that means first sequencing was takes place in 2002 by the scientist Cello and its co-workers in the laboratory, and first synthesized organism can be developed in this 2002. In 2003, again genome of this bacteriophages was assembled for 2 weeks for the James River Institute. See, this is what is this figure? This is the bacteriophages, which can also be kill bacteria, and this is a structure of a viruses. So these bacteriophages are also synthesized or sequenced by in 2003. So application of synthetic biology. So this is the main important part we are going to discuss. The ultimate goal of synthetic biology is to develop commercial application that will benefit the society or our needs. For developing countries or developing needs, developing populations, large number of needs for the economically and society for benefit of the society, this synthetic biology can be utilized. Here are some examples. To design and build engineering biological systems and process information is also utilized. Manipulate chemicals, for example, fragrances the essential building blocks for a wide range of chemical products, detergent, cosmetics, perfume, industrial lubricants, all these things can synthetically engineering designed by yeast. That are the chemicals which can be produced by this yeast. So maintain, enhance human health and our environment, engineering yeast to produce estimatic acid and anti-malarial drug. Researchers and companies are working on way to produce vaccines through synthetic microbes. So like in coronaviruses, the vaccines can be produced in this type of synthetically designed corona vaccines. So See, these are the different applications of the synthetic biology. Fabricate materials and structures, re-engineering the type of the screen systems, and Somela trypsinium are the create spider linked protein. So these are the different examples where you can be. We are going to utilize to this synthetic microbiology or synthetic biology for our needs. Then produce energy. This is the important for the gain energy. We also the synthetic biology are going to study due to it also meets the need of the energy demand of the population. So some organisms which are synthetically produced, they have to utilize for production of energy. Organisms which are genetically engineered to break down the biomass into sugar into fuel. Because nowadays we already discussed biofuel, biogases, biomass. These are mostly the future and green energy resources, and that can be converted, the biomass can be converted into biofuel or ethanol through the degradation of this compound into ethanol by some microorganisms. So these microorganisms which are either aerobic or anaerobic, we already discussed all these things. So the efficiency of these microorganisms, the degraded part of the microorganisms that are naturally occurs microorganisms, they may not be sufficient. So we may design some new synthetic microorganisms that can easily degrade this biomass into ethanol or fuel, biofuel. Some algae are naturally produce oils, but through synthetic biology tools, algae can be re-engineered to produce oil that are chemically similar to the oils that are currently today's transportation infrastructures. There are some examples. Algae can produce oil, but nowadays the synthetic biology utilizes genetic engineering to produce some synthetically restructure of the DNA of this particular algae that can produce that oil which is resembles the oil such as petrol and diesel. So these are the utilized for the biofuel productions. Natural production product substitutes. So synthetic biologists are also working to replace natural products with synthetically produced equivalents. Naturally something is produced is very much taking longer time and the production of yield is also very low. But synthetically we are designing some organisms or that can be yield capacity is very high and produce within low with minimum time. For example, the poultry or chicken which are nowadays having grow within 3 weeks or within 2 months or 1 month they can grow from 1 kilo to 3 4 kilos. So these particularly growing of the meat production that can be help for the meat production. Another example is the milk production. If some natural cow and buffalo giving 2 3 4 liters naturally, but nowadays due to this genetic engineering the cow buffalo gives 30 40 50 60 liter per day. So this type of production of can be changing the naturally to synthetic products. So that can be also utilized. Synthetic production rubber through isopropene, crucial building block for making artificial rubber. Artificial rubbers are also produced by these processes. Gene encoding for isopropene is found in rubber tree has been synthetically engineered by E. coli to produce isopropene. This isopropene can be produced by actually this rubber plant, rubber naturally found in rubber, but nowadays the E. coli bacteria that can be also produce isopropene by thus modifying the gene. Other products currently producing through synthetic biology includes vanilla, stevia, palm oil and other things. These are also synthetically produced nowadays in laboratories. Previously, only natural sources the way where can we get, but nowadays we can laboratory synthetically produce these things. These are the different products nowadays. Yeah. You also utilize this product day by day in our natural life. Impact of synthetic biology on the conversion of the sustainable use of biodiversity. So somehow we are going to utilize this the synthetic biology technology, but it also impacts some consequences or some having some things that can be also for the use of biodiversity. It can impact some things. So synthetic organisms threaten biological diversity. Natural things are going to be modified. So we are going to disturb the natural chain or natural process. So that the natural process is going to be threatened. So the natural species are going to be extinct. So biodiversity loss or for the environment, this is the main consequences of this synthetic biology. Other internationally or internationally from a lab. That means, for example, this pandemic that have been the coronavirus pandemic is also due to this. You have heard this in Yuan, in China. Some are experimentally designed some new type of viruses. That is nothing but the synthetic biology applications. So it may be a vast disaster in the world nowadays. So this may be a threat to our natural system. So organisms are belonging synthetically engineering, survive, function and propagate natural environment. And the actually synthetic biology are utilized for our some benefits, but it can replace the natural things and they can adapt the natural processes that compete with the natural things. Regulation of synthetic biology. So by looking of the risks to the threats of biodiversity, the government or some international agencies, they have also has some rule regulations on the synthetic biology. So risks of synthetic biology to pose in human health and environment is serious since synthetic biology ability to create organisms and have never existed before or of the complexity, because we are synthetically organized or design a new organisms which may not be present naturally. So this may cause a different disasters for the human species in futures also. Best examples is your live example is coronavirus. So we must establish proper regulation to safeguard this technology and for before too late. That means you have to matlab, regulations. We have to make some regulations to develop the synthetically organisms, structure organisms or biology for this uncontrolled use. Because this synthetic biology is a threat to biodiversity and human beings if it is uncontrolled. So let's see some different aspects of the synthetic biology. Current application of synthetic biology. What are the different applications of current applications? So in summary, current application includes food and agricultural applications. Food and agricultural application, most of this I have already given examples, that is production of meat and production of cow. That is both are nowadays for agricultural needs. We are going to modify this genetic of some organisms to production of this food and agricultural needs. Microbial based fertilizers improve the soil health, enhance the crop production, swapping mixture and nutrients, suppose surfaces oil from pathogenic diseases in crops, maintain the symbiotic relationship. These are the microbial based fertilizers. So we are going to apply these to enhance the crop productions and the enhance the disease resistance variety, maintain the relationship between these different species. Then genome edited crop and higher yield to resistance disease and involve nutrition. So because to gain higher nutrition value, we have to going for this resistance variety. Establish nitrogen fixation mechanism of non-legume crops. We already know the nitrogen fixation is done by the nitrogen fixing bacteria, which is only found in leguminous roots, noodles of the leguminous roots. But nowadays, this non-leguminous roots, you can incorporate this synthetic production of materials, organisms that may be capable of doing nitrogen fixation. This is very much good. A array of genotype editing technology also used. Agri food applications includes golden rice project. That means golden rice means the color of rice is not gold, because of the production of rice production is boosted in a very rapidly manner. So that called golden rice project. Means initially in 1 acre land if rice production 35 quintals, nowadays this boosted to 35 to 70 quintals. That means it doubles the yield from the same field. So this is called golden rice production field. This is already implemented in different regions. various plant-based, cell-based alternative protein products to replace the energy resources incentive the animal meat. Already discussed, to grow the meat, because in natural processes the meat will be growing, it takes time, but due to this genetically modified or this synthetic application of synthetic biology, the energy, actually food and agriculture, these are the nothing but the source of energy. So this energy demand can be met by this food and agriculture system. Then different environmental applications, electronics waste recycle, this is a emerging new uh, types of waste, that environmental solid waste, which can be called e-waste. So the use of this engineer microbes to explore the recover the metal and rare earth element to produce on cyclical electronic suites. That means the electronic suite, if your mobile or laptop which will be going damaged, or you just throw out in dustbin, so it cannot be biodegradable. And for making of these electronic applications, it, there are so many rare earth elements, so many things, silicons, semiconductors, all are used for the making of the electronic chips. But nowadays, genetically modified these applications of synthetic biology, some organisms, they can help to degrade or separate different kinds of elements that are present in these electronic chips. Also, second important aspect is the plastic. Plastic is the main headache of nowadays in our environment, because the plastic is non-biodegradable. So it is remain in environment in our soil for thousands of years, but it is not biodegradable. So production of biodegradable plastic is the most important features of the nowadays, which improve the recyclability. By plastic that can be decomposed by these microbes due to this synthetic application of synthetic biology. We are genetically modified, or we are going to be synthetic some microbes or some organisms that can be going to decay this waste plastic to non-biodegradable to biodegradable. Reduce the generations, discovery of the polyethylene or the PET degrading bacterium. That means the polythene, we are going to say the plastic is called polythene, normal our local language, polythene bag de do, so polythene, so the polythene is called polyethylene, polyethylene tetra terephthalate. This polyethylene or the PET, that is called plastic, actually the chemical name, and going to design some bacteria or some going to modified, genetically modified organisms which can destroy these things. So isodenal succulensis is the two key of enzymes. So we are defining different enzymes are also produced more efficient breakdown of conventional plastics and fight against the plastic pollutions. So these help to reduce the plastic pollution. So this is the uses of synthetic biology. Another applications are bioremediations and biosensors. Bioremediation is nothing but the utilization of the microorganisms or some things that can be going to absorb the toxic materials from the soil, and it can reclaim in the soil. So from the soil, the toxic pollutions or pollutant can be intake or can be degraded by these microorganisms. So naturally the microbes are not going to degrade these toxic chemicals, but if you are synthetically sequencing some DNA, and due to the help of genetic engineering and the synthetic biology, if you are design such a bacteria or such a microorganism that can also degrade these or toxic pollutants, or even if some oils, because sometimes oil spills occurs in the soil, then the soil fertility get lost, because the soil never get degrade in the soil. If you are designing some bacteria that can also degrade the oil also and reclaim the oil, that is very much useful. That is called bioremediation. Microorganism to even plants also engineering to degrade pesticides and remove pollutants. So chemicals, pesticides, fertilizers, which are toxic chemicals which can also degrade by these microorganisms developed by synthetic biology. Biosensors, that means a large number of uses including production of photographic bacteria. They can also be developed toxic chemicals such as arsenic, because sensor means they are going to indicate something. If in your level the arsenic pollution is very high, arsenic level is groundwater is very high in that particular area, So some bacteria are found, they can indicate that without testing, going to the laboratory for testing whether arsenic content is high or not. They can indicate by changing the color or by having some properties that we can know that this area having higher levels of toxic chemical and arsenic, all these things. So this genetically modified or this bio synthetic biology applied in this field also. Next application is industrial applications. Industry we already discussed. We are going to synthesis different kinds of drugs, different kinds of vaccines, different kinds of all these things, proteins. Uh, so in industrial advanced materials, advanced manufacturing process, the synthetic biology is also applied. Stimuli response functional polymers produce protective clothing and these buildings. So these are also the different uses. Programmed protein that can self-assemble into predictable shapes and used in chemical and medicinal industries for designing a particular drugs or particular kind of things in the industry. Cell-free manufacturing case of downward processing of the scope of the productions. Waste to energy projects that waste renewable sources that can be alternative feedstock for the production of energy, because we are going to throw these garbages or going to throw those waste that is not utilized anywhere, but if you are utilizing this process or synthetic biology to generate energy from this waste, that is very much helpful. So nowadays this is also going on. Sustainable products and biofuels, these are the most advanced things in our system for the application of this synthetic biology. Mushrooms, leathers. Nowadays actually the leathers which are you have a daily day to day you are uses like vanity bag or the belt or many things that are produced from the leathers, that are actually not come from the natural leather of a plants, natural leather for any types of animals. Nowadays leather can be synthesized in plants or chemical factory synthetically. So these leathers are also produced by this main applications of this synthetic biology. Spider silk, lightweight threads, these are the different products that can be utilized, main application of synthetic biology. CO2 based photosynthetic pathway to produce chemical from the cyanobacteria. These are also applications, CO2 reductions, and where we know that carbon dioxide is the most evident greenhouse gases found in environment. So the level of carbon dioxide increases, then obviously the global warming, climate change, all this happens. So how this carbon dioxide can be reduced? So due to the synthetic biology, we are utilize this some kinds of bacteria or some kind of application microorganisms that can be capture this CO2 from the atmosphere and reduce convert this CO2 to oxygen. That is very much helpful for this, but this is not in large scale. We are going to be utilizing or maybe in futures. Biofuels, that means photosynthetic production of biofuel using genetically engineered microbes. We already discussed the biomass can be converted into biofuel through the medium of this genetic uh, synthetic biology applications of the microbes which are synthesized in laboratory, which may be helpful to needs of this change uh, production of this biofuel or energy. Here, algal biomass are converted into bioelectricity, bioethanol, biodiesel, biochar, biofuel. We have already discussed all the things. What is biofuel, biomass? Next application is medicines. So design of some proper medicines and vaccines we already discussed. Diagnosis and prophylactics and pharmaceuticals. In these cases also the synthetic biology are applicable. Thyrokinetic cell sensors, activators that sense the disease that produce the responsible itself. That means diagnosis of some diseases that can be also done by these synthetic applications. In vitro diagnosis of live engineer bacteria to detect and record the pathogen of the different antibiotics. In order to test some drugs, some vaccines or some things, we can also utilize this synthetic biology for these purposes. Pharmaceuticals means to produce small molecule drugs or vaccines or medicines in the help of this synthetic biology or synthetic modified, genetically modified these organisms which can be produced. Small molecules fight against cancers. So this remedy of cancers can be also found with the help of this synthetic biology. Usage of microbes to synthesize demanding small molecules like these are the plant sources. This means so different produce different kinds of medicines, drugs. Synthetic biology is utilized. Therapeutics mean to treat different kinds of the organisms genetically engineered viruses to correct defensive genes, all these diseases. If you are having some diseases that can be also cured by this synthetic biology, such as we are developed last slides we are going to see the bacteriophages, which is the main produced by the synthetic biology. Bacteriophages, well, this one, this is the bacteriophages which is developed in 2003, and these bacteriophages can be uh, going to cure HIV and AIDS. If you know there is no cure for the HIV and AIDS for these things, but nowadays this HIV and AIDS can be cured with the help of these synthetic biology applications. HIV, cancers, these are the rare diseases which are very much common nowadays. So, but there is no remedy at all. So, these can be in developing stage, and this may be cured by therapeutics of this application of medicines. Future trends in sensitive biology. So, this is going to be more applicable. So, you know, not going to in details, but just to know regenerative medicines, the medicines we are going nowadays. These are the in future trends. Future trends means we are. This is in scope of these futures. How the synthetic biology can be applied in future. First thing is regenerative medicines. Some medicines we are going to take, but after some times, what's happen? The medicine is going to excrete with our urine or with our stool. But some medicines are going to regenerative. If you once take the medicine, the fundamental uh, principle is if you once going to take the medicines, it remains in your body, and even if there is whatever in futures the disease comes or the pathogen comes, it kills you the pathogens that can be effectively. So there is no need to take the medicine repeatedly after 1 year 2 years when the disease again and again happens. So this is called regenerative medicine. This is also in future scopes, making xenotransplantations a reality. Xenotransplantations a reality. That means this is the again new types of application of the synthetic biology or genetic biology. That means if your see the figure, what is the figure? The one is called human, another is called this sheep or any other organisms. If kidney of a human beings is damaged, then we are going to replace the kidney with some donor or if you are available. But due to application of the synthetic biology, we can replace this kidney from the different organisms or different animals through these types of application or xenotransplantation. This is also going to in futures. Then 3D bioprinting, this is a very much high genius techniques. If your heart transplantation or if you need a heart, going to print the heart with the application of the synthetic biology. This is a bioprinting. So this is not in process nowadays, but in future it may happen. Likewise, when you are going to need some photocopy, just going for nowadays for printing of photos, and if you are having some designing, then going for a 3D designing, just 3D block printing. But 3D bioprinting is something very much advanced techniques with the help of this biosynthetic biology. If you need some organ or body part, just give with the help of this synthetic biology, just print it and just fix it. Then nano robots, these are also in future aspects of the synthetic biology. Cancer can be cured with the help of nano robots. The robots which are small, very much in nanoscale, and that can be cure any type of disease like cancer that one implemented in your body. This is in Hollywood pictures we have seen these kinds of ideas. This is not so much far now. It is like a movie, but some after some 10 years, 20 years, this may be reality, not in is in near future. Then future vaccines. Future vaccines means whatever diseases that can be comes, we are going to take vaccine from one disease another diseases, so different type of diseases we are going to take. So if you are design some type of genetic chips that can be implemented in your body which can cure all diseases, we become disease free. So these are the some things that going to be implemented in near future. Synthetic mushrooms, sorry, synthetic chromosomes, that means the modified genetic modified or some chromosomes. If you, this is also possible if it is a possible to change the chromosome, then there also cure some genetically disease or genetically inherit disease in our body. So these are the applications of synthetic biology. And potential risks of synthetic biology, we also know whenever there is a advantages, there must be disadvantages, and we are already discussed the disadvantages such as biodiversity threats on biodiversity, then genetic mutations. Next is developing of new viruses like corona pandemics. This is also maybe possible. So these are the different risks of making, because we are producing new kind of organisms that never existed before. So that we don't know whether whatever, if we are going to accidentally produce a high risk organisms or high risk viruses, bacteria that may kill whole humanity or whole organisms. That also risk is there. So going to the risks, we are going to assess the risks, and we are going only see the benefit part of this synthetic biology. Obviously environmental risks, biosafety is there, that means environmental health is verified. Ethical issue, yes, this is a one different thing. Ethical issues, ethics means actually we are going to decide whether we are going to do right or wrong. If you are, we are going to take some steps. For example, if you are going to study some higher studies in some branches, or you are going, you should not decide or that either it is going to do benefit to society, benefit to you, or whether it is going to harm to society or harm to you. So this synthetic biology is also this type of dilemma, because if you are utilizing the synthetic biology for the sake of the or the benefit of the world or the danger side of the risks of the world, because we may not know some kinds of, you see in Hollywood pictures they are utilizing these bacteria and viruses or new genetic new type of things that may kills whole humanity, that may be possible. So it is a cause of discussion whether it is good or bad. But this is called ethical issues. Some people say it is, oh no no it is good. Some people say no no it is bad. There may be a risk. So where we go? Whether we go for right or whether we go for wrong. So this is mainly discussion, class of discussion. Then creation of unpredictable microbes. Obviously, we are not know whether these microbes are not pre-existing before whatever we are going to studies that already existence in the world. But if you are not, that is alien species which cannot be in the from the world, you are making by mistakenly some more strange alien species that may be a risk to our environment or total humanity. So biology from laptop, that means it will start from DNA sequencing and end from the new organisms which can be applicable in different things. Then regulation synthetic biology already discussed. There are so much different types of or things that can be going to regulate. Here are some regulatory authorities here. It is going to permit us. The risks of synthetic biology of human health or environment are serious, since synthetic biology has ability to create new organisms that never existed before, and the complexity will increase over time. This is the risk, because we not do not know what is the impact of this new type of virus or bacteria that may we are risk, very risk to the environment. So safeguard must be there. So there are different authorities are there, like Organization of Economic Cooperation Development, European Academic Science Advisory Council, Presidential Commission of Study of Biological Issues, USA, German Central Committee of Biological, European Academic Science Advisory Council. There are some advisory council. They give some rules and regulations for the working in the field of the synthetic biology. This is a very critical issues and very critical. Not is very simple. So whatever maybe we are going to do, that is for the sake of the goodness of the well-being of the human or the environment, not for the bad for the environment, all the things. So these are the some applications of this synthetic biology and synthetic things. So if you have any doubt about synthetic biology, because this is somehow who works or who are in a branch of life science, they may better understand what is this genetic engineering and biomolecules, DNA, RNA sequences, how it can be done. Any questions? No.
No, sir.
Okay, then next class we will going to discuss about this genetically modified organism, which is what is GMO. That is also one part of the synthetic biology, and what are the importance of this GMO. Next class we will discuss this one. Your syllabus for unit 3 is going to be end after two more classes, one GMO and another hybrid system. So next two classes it is free, unit 3 and unit 4 another 5 classes. Okay, if there is no class, no questions, then see you in the next class.
Thank you, sir.